Hello, I'm Zahra Shah Mohammadi, a master's student in molecular medicine at Essex University. Today we want to discuss about design and validation of an expression vector for recombinant tau Fab expression in mammalian cells.
The gene of interest in this study is tau antibody Fab gene, and this project aims to generate a recombinant vector carrying the tau antibody Fab gene. The antibody fragment will be expressed in mammalian cells, purified, and functionally analyzed. Tau stabilizes microtubules in neurons, but in disease like Alzheimer, it forms toxic aggregates. The tau antibody is used to detection and neutralization of these proteins. The pcDNA 3.1 plasmid was chosen for our eukaryotic study and its optimization in mammalian cells to ensure proper expression and folding of the Fab fragment. Its strong CMV promoter enables high protein production, essential for analysis and purification. The plasmid's restriction sites were verified for gene insertion compatibility, and it carries ampicillin and neomycin resistance for the selection in the bacterial and mammalian cells. In addition, I also examined the restriction enzyme sites within the gene of the interest to ensure that the enzymes would not cut within the gene itself, preventing the loss of any critical sequences. As you can see here, the BAMH1 and ECOR1 are the selected restriction enzyme that we design our primer based on these two restriction enzymes, and our gene of interest inserted into multiple cloning site of the plasmid. In this slide, you can see the DNA sequence of the tau antibody. Based on the top strand sequence and the selected restriction enzymes, the forward and the reverse primers were designed. As you can see here, for forward primer from 5' to 3', first we have the leader sequence, and then we have BAMH1 restriction site for forward primer, and then as it is a eukaryotic plasmid, we choose to have Kozak sequence. After Kozak sequence, we put the gene of interest. In the reverse primer, as you can see here, from 5' to 3', we have leader, and then we have ECOR1 restriction site. After ECOR1 restriction site, there is a stop codon, and as we wanna purify the protein at the final stage, we put histidine tag here, and after histidine tag, we have our gene of interest.
There are four main stages for designing and validating this expression vector. The first one is about cloning, in which the gene of interest amplified, digested with the restriction enzyme, and ligated into plasmid vector for transformation into bacterial. The transformation is the stage in which the recombinant plasmid is introduced into competent E. coli cells for replication and selection. Transfection is the stage in which the recombinant plasmid is delivered into mammalian cells to enable protein expression. And the final stage is about post-transfection analysis, in which successful protein expression and functionality were confirmed.
As mentioned, the first stage is about cloning. The tau antibody gene was amplified via PCR using designed primers, followed by digestion of both the selected plasmid and the gene with ECOR1 and BAMH1 to ensure compatibility. After confirming fragment sizes via agar-gel electrophoresis, the purified gene was ligated into the vector at the MCS under the CMV promoter. The use of two restriction enzymes ensured correct orientation and prevented misligation. Followed ligation, positive clones were validated by restriction digestion to check fragment size. Finally, Sanger sequencing was performed to confirm both the correct gene sequence and orientation. For sequencing, universal primers were used. As mentioned in the picture of the recombinant plasmid, the forward primer was designed from the CMV promoter region, while the reverse primer was placed near the poly A signal, ensuring full gene coverage in sequencing. In transformation stage, the recombinant plasmid was introduced into E. coli XL1 below were chemical transformation. After heat shock and recovery in SOC medium, transformed bacteria were plated on Luria bertani agar with ampicillin for selection. Only colonies containing the plasmid would survive. Colonies were ground overnight and plasmid DNA was extracted via mini-prep, allowing for verification through restriction digestion and agarose gel electrophoresis. These processes enable confirmation of successful gene insertion by comparing fragment sizes with the expected ones, ensuring the correct vector has been engineered. Final step of cloning is transfection into mammalian cells and selection. The tau antibody gene plasmid was transfected into HEC293A cells, as we choose them as our cell line in this project, using lipofectamine. The DNA lipofectamine complex facilitates plasmid uptake, leading to gene expression. To select successfully transfected cell, G418 neomycin was added to the culture medium, ensuring only plasmid containing cells survived, as the pcDNA 3.1 vector carries the neomycin resistance gene. We mentioned about it in some previous slides. For verification of functional expression, Western blotting was performed to analyze protein expression. An antihistidine tag antibody was used to detect the presence of the expressed tau Fab fragment, confirming that the protein had the correct size and was expressed successfully. Additionally, the expressed protein was purified using nickel NTA affinity chromatography, which specifically binds to the histidine tag attached to the antibody. This step ensures the isolation of the correctly expressed antibody for further analysis. Thank you for your uh, concentration and your time, and you can have the reference at the final slide.